Hi, everyone. Thank you for coming to the harmful algal bloom session. So, yeah, I'm Cary Lopez. I'm with the Florida Fish and Wildlife Research Institute. My co-chair for this session is Courtney Schmidt, and she's on virtually. She is staff scientist with the Narragansett Bay Estuary Program. I'm not sure if she can say hi, but she'll be on virtually and moderating for comments in the chat. So in the HAB session today, we've got 6 speakers, and the topics we'll cover are how research, novel tools, modeling and monitoring can inform management of the major HAB taxa impacting Florida estuaries. So we'll start the session off hearing from Patrick Schwing, who is the assistant professor of marine science at Eckerd College. He will be talking about investigations on Karenia brevis blooms and benthos coupling in the coastal West Florida shelf region.
Good afternoon. Thanks, Cary. Thanks for having me. So yeah, I'm going to be basically giving a little bit more information about an ongoing research collaboration that we have with Fish and Wildlife Research Institute, FWRI, essentially coupling the seafloor of the benthos to an ongoing harmful algal bloom monitoring project. I just want to acknowledge all of my co-authors here. This is the work of many people, many of who are undergrads at Eckerd College. So a bit of an introduction here. First and foremost, most of the Karenia brevis research has been done in the water column in the neritic zone. And we don't know all that well what the role of the benthos or the seafloor is in initiation specifically, and also in termination of the blooms. We also don't know a whole heck of a lot about the seafloor community impacts from harmful algal blooms. So just recently there was a paper published in the South China Sea looking at the possibility of resting stages in a similar organism to Karenia brevis. And so again, it calls into question whether the benthos has anything to do with initiation and termination. Bob Weisberg, who will be giving a talk in just a few minutes, also published a paper with colleagues in 2019 and had, through physical oceanographic methods, identified a potential initiation zone on the West Florida shelf. So we are providing a lot of surrounding ancillary information, not performing specific counts on Karenia brevis, but all of the other information to help guide those estimates. So providing sedimentology to provide an environmental context. We're also looking at forams, benthic foraminifera, which are very useful bioindicators of benthic health or seafloor health. And then last but not least, we're also taking short core records and now hopefully longer core records here in the next few weeks to look at the historical presence of Karenia brevis in sediments as well. So all 3 of these together will help us progress our understanding of initiation, termination, and also the impacts to benthic health. So just to familiarize you with the setting, I'm going to be focusing almost entirely today on the foraminiferal story that have been collected along the Rome transect on the bottom left map there. So just to orient you, these are 5 routine monitoring sites ranging from Rome 5, which is the closest inshore, out to Rome 1, which is the farthest offshore. We've collected surface sediment grabs there for foraminifera and sedimentology on a monthly timescale from January of last year up to February of this year now, and we'll continue that for at least another year. We're working on deploying sediment traps in these areas to work on fluxes from the sediment column. We have collected short sediment cores to just for proof of concept, and we're planning on going out to collect more of those in a few months. And last but not least, bottom water CTD measurements for environmental context. All right, I'm not going to go through all of these bullet points. I realize this is a lot of text. So we primarily use benthic foraminifera as tools. In this case, they are very useful seafloor indicators of health. So they're single-celled protists. They create their shells out of calcium carbonate, so they're nice for repositories and sediment records, so histories. They're widespread across marine and also transitional systems. They are also extremely diverse, so there's 4,000 different extant species currently. What's really useful about them is that those species can be categorized into groups of very sensitive to certain pollutants or contaminants to very opportunistic, and they can be calibrated as such. So you can actually calibrate them for a specific problem, in this case, harmful algal blooms. All right, so some preliminary results here. Just for context, in the top right, I have the map here. This is a multi-dimensional scaling plot just to see how the assemblages of benthic foraminifera break out at these sites over time. So the shaded polygons, the larger they are, basically tell you how variable that site is over time. You'll notice that Rome 5 on the right-hand side has the largest polygon. It's the closest to shore, has the largest temperature flux, oxygen flux throughout the year. This makes a lot of sense. Rome 1, 2, and 3 in the middle of that plot break out with depth. There's no surprise there. As you make your way offshore, this makes a lot of sense. And then we've also got a couple of things here. So Rome 4 actually breaks off on its own on the bottom left with both fluorescence and oxygen. So we think there's a primary productivity story here. Again, just scratching the surface as to how this all breaks out. So looking at the time series here, I'm going to try and unpack some of these plots for you. On the top, we have abundance, so basically how many foraminifera are at each of the sites over time. The second plot is richness, so how diverse that community is over time. And on the bottom, we have the AE, or ammonia alphidium index. Essentially, when that index is high, oxygen is low. So first and foremost, this is the first time we've demonstrated monthly temporal variability in benthic foraminifera assemblages on the West Florida shelf. So we know that they're appropriate for things like harmful algal blooms in monitoring. We see the highest abundance at 3 and 5, so you can see a few peaks up there on the top right. And then I've also broken up, this is over the course of January 2021, January 2022. The yellow, orange, and red correspond to the FWRI's ranking of low, medium, and high cell counts. This is not site-specific, this is a regional coverage, but just for a rule of thumb. And then we also see increased richness, so increased diversity offshore from April to October at 3 out of the 5 sites. And then as of October, where we have those high counts, we see a decrease. So this could be seasonal, this could be harmful algal bloom related. Again, we need a little bit more information, a few more years of collections to fully calibrate this. Let's see. So one of the tools that we're developing using this time series is a marine biotic index. It's a commonly used marine biotic index, especially in Europe for the European Water Framework Directive. It's based on the AZTI Marine Biotic Index, or AMBI. We populate that marine biotic index with foraminiferal assemblage information, and we essentially construct ecological quality status, or benthic health measures, from 1 to 7. So it's a very easy to digest, easy to understand index. And for some of the reasons that I just mentioned, forams are particularly useful to populate an index like this with. Last but not least, we can assign each species to a certain category based on how sensitive they are to a specific stressor. And so in this case, we're calibrating it to total organic matter to start off with, total organic carbon. And then eventually we'd like to tailor it to either cyst counts, cell counts. You can also tailor it to other questions like ocean acidification and pH. So this is some preliminary data. This is hot off the press. This is a composite from January 2021 to 2022. The colors are reflective of a high status being low in blue or purple, all the way up to a bad status in red. And you can see, again, we have a variability from site to site and with time. So we think it's working. Additional validation is needed. We're going to be adding a lot of data to this in the coming months, and hopefully it'll become more and more robust as we go along. Looking at that index over time in the same context as those first plots with low, medium, and high counts, there is a little bit of variability between site. Obviously, the one closest to shore, Rome 5, is that orange plot, that orange line up towards the top, has the highest AMBI ranking, the lowest benthic health. The others that are farther offshore get progressively more healthy according to this index. What's really interesting, if you look again at that October timestamp with the red rectangle, you do see an increase in the AMBI score at 3 out of the 5 sites. Now, again, we can't definitively say that this was caused by the harmful algal bloom yet. We need to, continue to work on the sensitivity test as well, potentially calibrating with cysts. Some broader applications of this approach. These are just gradient maps of all of the other data products that we produce with forams, along with sedimentology and radiochemistry. We kind of used the Piney Point effluent release as a way for us to get out there and set some baselines. We were not able yet, at least, to assign any specific impact and response from the forams in relation to Piney Point. But now, because of that effort to go out and take all these samples, we have some baselines for most of Tampa Bay and the inner West Florida shelf around Tampa Bay. So Becca Larson gave a talk this morning about this effort, so feel free to talk to either of us if you guys are interested in any of this work. All right, so some of our broader goals. Right now, we're setting spatiotemporal baselines on the seasonal to interannual basis. So hopefully about a 3-year baseline period, we'll actually have a formidable biotic index to judge impact from harmful algal blooms. And then also thinking a little bit more broadly, we're going to take longer sediment cores shortly to expand that to a decadal to centennial scale record of the magnitude and intensity of harmful algal blooms throughout the last 100 years or so. So that'll give us some benthic health estimates during that time period associated with HABs. It'll also help us establish pre-anthropogenic baselines for harmful algal blooms. So we are currently in that effort working on seismic lines offshore to identify depot centers of sedimentation, and going to go out and collect those with a vibrocore system over the next few months. We're applying more robust statistical methods like Canico and others to try and disentangle some of the other environmental parameters in addition to HABs and their effects on the seafloor. And we're going to continue to work on our biotic indices as well. Last but not least, and probably most importantly, is we're working with our collaborators at FWRI, USGS, and the Royal Institute of Oceanography in the Netherlands to add cyst counts, to add other biomarkers and molecular indicators of HABs to these records. With that, I want to thank the HAB group at FWRI and thank you all for your attention and ask you guys if you have any questions.
Questions. If you have a question, raise your hand and Joe can bring you the microphone.
Thanks. Hey, Patrick. That's really good work. I just had a really quick question. One thing that I took away from this is the differences between Rome 4 and 5 seem to be pretty large, especially that one graphic that you showed with the AMBI score, and you had like Rome 5 way up here, Rome 4 up there. But on the map, it looked like they were pretty close together. They weren't that far apart. I was wondering why the big difference relative to the fact that they seem to be pretty close together. Thanks.
Yeah, that's a really good question. So looking at the AMBI scores, the kind of color plots with month, Rome 4 seems to be in a sweet spot. And I don't really have a good answer really. 5 is closest inshore. We see a lot of variability in a lot of environmental parameters. So just kind of peeling out HABs from all those is going to be difficult. 4 remains pretty healthy the entire record. And then you see more variability offshore. So I don't know if it's 4 is in a location that's more stable. I need to look into that. But Rome 1, for example, the farthest offshore should be the most stable seasonally. We see decent variability. I don't know if you noticed, but it went from a purple to a red, on a monthly scale. So there's quite a bit of back and forth. Again, we're just scratching the surface. So hopefully over the next, few months, we can have a little bit more definitive answer to that question. Yeah. Let me off easy.
Okay.
All right, thank you very much.
Thanks, Patrick. Great talk. Okay, so now we will hear from Dr. Chuck Jacoby, supervising the environmental scientist for Florida's St. Johns River Water Management District. And Chuck will be talking about using Geocollaborate to integrate and communicate data on harmful algal blooms.
Thanks, Cary. So yeah, my job is to set the stage, as it were, the context for this work that we got funded from the DEP for an innovative grant. And then I'll turn it over to Virtual Dave, who will walk you through some of the things about Geocollaborate. So what we're going to be talking about is the Indian River Lagoon, which is a system of 3 water bodies that are connected by canals, spans about 40% of the East Coast. And they're receiving water bodies. The lagoon has had some issues, and one of the key ways you can tell that is by looking at seagrasses, because the lagoon should be a seagrass-dominated ecosystem. This shows you aerial extent, which is the green bars, that's acres, and that's the left-hand y-axis, and then the right-hand y-axis is mean percent cover, that's the black line. The take-home message is there by the red arrow. That since 2009, we've lost about 58% of the acres and about 89% of the cover. So that leads to other effects such as hungry manatees. Why did we lose that? Well, a variety of phytoplankton blooms shaded the seagrass. This is a picture of the 2016 brown tide, which is the focus of a lot of the work we're doing with the Geocollaborate tool. But we had a series of blooms that first started in 2011 with what at that time we called the super bloom because it was of the highest intensity and longest duration of anything recorded in the previous 20 or so years. But since then, we've seen more intense and longer duration blooms, including the 2016 bloom in the middle. And sometimes besides shading, these blooms would crash, and the crash led to low DO and fish kills. And 2016 was one of the largest ones recorded. So if we look a little closer at that one, we can see here from these satellite images, again, satellite imagery modeled chlorophyll with the hotter colors being more chlorophyll. The bloom started gearing up in late November. By January, it was pretty much at full swing. In February, it was carrying on. And then sometime between sort of middle of February and into the middle of April, it crashed. The hassle with satellite imagery is if there's clouds, you don't see it. And that was one, partly what led to the crash, and two, made it hard to see. But we had sondes in the lagoon that recorded things like chlorophyll A. So this is a graph of chlorophyll A through time. And again, that red arrow points to where the chlorophyll A dropped. That was when the cloudy days started, the bloom crashed. Following that, you get low DO. And again, the red arrow points out where it drops below 2 milligrams per liter, which stressed out the fish, caught a fish kill. The yellow ovals just point to some concerns about using raw data. With something like the tool we're going to talk about, you need to be careful. You need to understand how much you can trust your data. If you want to talk about that, you can talk to KJ, who's over there. So the context, the overall context, in 2016, we probably weren't as prepared as we wanted to be. We learned from that and started holding weekly meetings with people exchanging information and trying to look ahead and see what we needed to worry about. Fortunately, in 2018, which was another very intense and fairly long-lived bloom, we escaped a big fish kill. It was a better behaved bloom and it just gradually faded away. 2020, likewise, we had another bloom and also escaped. But the conclusion was that we were looking for a better tool to exchange information, keep everybody on the same page, and maybe even look forward and predict where we might need to take actions. And that's where Geocollaborate comes in. And I don't know if I'm going to be able to make this run.
Geocollaborate brings a new capability that enables real-time active locations. Please inform Dwayne. I'm going to draw this polygon surrounding two areas here. And once it is drawn, I'm going to save that as a separate layer. And now it can be a data layer that's queried. Notice that it's displayed rapidly in the follower's instance. We'll display it on every follower's instance and also appear in the dashboard. This polygon can be queried. This is a collaborative message, real-time environment. Here's an example of how the media can use the dashboard for a real-time reading. So you have the data sets that are loaded here. They have full control over what they want to show. Let's say they want to show these two dots with fish kill photos. All they have to do is go into other functions, show full screen. They can move things over to the side like the legend. And so if they want to see an image, there it is. They have done no modification to the scientific data set that has been shared.
So the ultimate take-home messages is, we have access to data. Certainly, if we put our heads together, we could probably find other data that we would chase down. For example, if we'd have thought carefully, we might have in that 2016 period looked for cloudy days, looked for forecasts of cloudy days, which are out there. What we need to bring those data together, we need to understand how much we can trust each of these types of data and how best to use them. Generate the insights by combining the variety of data, and this is one of the tools that may help us do that better. And then again, benefit from something besides sharing slides, especially that 24/7, 365 access for people who are interested in the situation. And I'm happy to take any questions.
We have time for questions.
He just ran that way.
Hi, Chuck. Thank you for the presentation. I do have a quick question. So I understand there's a lot of different portals and everyone is already loading their data in their, enterprise databases in-house. So just to clarify, this Geocollaborate would require uploading the data to another platform in addition.
No, it basically just accesses the data. So you don't take the data anywhere. You just access it. So if it's in the proper format, this go in and get it. And like example, the dashboard would update. So if something updates on a regular basis, the dashboard automatically updates. But you're not collecting the data. You're not downloading. You're not taking the data anywhere.
Okay, so the leader, it seemed to me that the leader was putting data themselves.
You select data sets,
Okay.
yes. But that can be done in a collaborative way during a session. So people can say, what if we looked at this and draw in another data set that's out there?
Okay. Yeah, I'm just, my big question was getting data from, cybersecurity is a big deal. How is cybersecurity handled in this if it's connecting directly to the other people's data sets? But it looks
again, the concept is that these people are willing to share their data.
Okay.
So the NOAA data sets are there to be used, and this is a way to use them.
Okay, thank you.
As long as it can answer as long as it can access the REST endpoint for any geospatial data, it can pull in that stream. So most data sets, when you access it online, you can access that REST endpoint. That's all we need. So, and a lot of the time we have to communicate with, hey, we're explaining to stakeholders, hey, we're using your data for this sort of way.
I think we're out of time, so maybe you can catch up with Chuck if you have more questions. Thanks, Chuck. Okay, so now we have Dr. Bob Weisberg. He'll be talking about Tampa Bay coastal ocean model applications. Bob is professor of physical oceanography at the College of Marine Sciences at USF. where he heads the Ocean Circulation Lab with emphasis on circulation of the West Florida continental shelf and its estuaries.
Thank you. So this talk will be half advertisement and half science. And so let me begin by advertising my co-authors and also my sponsors. So today I'm going to introduce what we call our Tampa Bay coastal ocean model, which is kind of a unique circulation model for this region. It's unique in that it downscales from the deep ocean, deep Gulf of Mexico, across the entire continental shelf and into Tampa Bay. And it does that with very high resolution, down to about 20 meters. So it includes Sarasota Bay, Tampa Bay, the Intracoastal Waterway, and all of the inlets that connect these with the adjacent Gulf of Mexico. Here's an example of sea level and velocity vectors on an incoming tide and outgoing tide. This is all available on the internet at the site that I identified, and it's available to the public. So this model has been run for the past 4 years on a daily automated basis with this information available to any of you who would like to access it. And we also distribute it to NOAA on their good site, so it's available to agencies to use it for any of their purposes. The model works, and so here's an example comparing a simulation with an observation at 3 different depths at a station in the main shipping channel by Port Manatee. We've taken the tides out, and what you're looking at is the wind-driven and density-driven portion of the circulation. And so at all of these 3 different depths, the model actually gets the observation very closely. Tampa Bay is an estuary. Estuary circulation generally has a net outflow at the surface, a net inflow at the bottom. We see the net inflow here very clearly. The closest observation to the surface was 4.5 meters down, which is kind of in a transition zone. So in the observation, we don't see the outflow very clearly, but in the model, we do. And here's a station a little bit offshore, and the agreements there are very good as well. So for typical everyday conditions, the model seems to have fairly good fidelity with observations. Here's a test of at an extreme condition. So this is an application to Hurricane Irma. Irma, recall, was in September of 17. The model had just started running then, so this was our trial by fire, and we passed. The model's been running daily ever since. And so on the right-hand panels, you're looking at just snapshots of sea level and currents at 4 different times as Irma went by. And I draw your attention to the white areas. That's where the bay actually dried out. And so this was observed. It's not made up. And so as the water was leaving the bay, there were areas around the periphery that actually went dry, and the fish were flopping around and whatever. What this says is that to have a model of circulation in Tampa Bay, you must have drying capability. Otherwise, the model wouldn't work. So TBCOM has that. So how well did it do in Irma? The right-hand panels show the red is the model simulation, the blue is the actual observation, and the green is the sea level predicted by tides alone at 5 different stations throughout the bay. And what you see is the model actually duplicated the set down in sea level very closely. There is a caveat, however. We had to elevate, we drive the model with NOAA winds, and the winds were anemic. And so we had to elevate the NOAA winds to match observed winds. The factor was about 1.6. So with that, the model worked very well. Without that, we would have underestimated the set down of sea level. The moral of that story is that you can have a perfect model, but if you don't drive it with good forcing functions, you're not going to duplicate the observation. So the winds that are available for Tampa Bay really need improvement. They're anemic, and they're anemic because they don't really include winds from inside the bay. It's mostly winds from the periphery the bay. So when NOAA assimilates these winds into their models, the winds they're assimilating are too small. All right, so let's go to the next application, which was to Piney Point, and I will get to red tides. Just have a little patience. All right, so what you see is the model grid superimposed on a map image. The red dot is where the Piney Point effluent was issued into Tampa Bay. And so we went ahead and put a tracer in the model, and we put the tracer in at the rate of inflow that we were given. And so the upper panel shows that inflow rate, which varied from day to day, but it started on the 31st of March and it continued through the 8th of April. And the lower panel shows the accumulated amount of tracer in the model and versus the amount that actually was input in nature, and the two overlay exactly. So the model actually worked, it conserved tracer, and so we can use that as a tool of figuring out where the stuff went. So here it is. Here's the first 10 days. Red is the highest concentration. Blue is the lowest concentration. It's a log scale. And so, you know, the traces spread out very rapidly. It actually spread throughout the entirety of Tampa Bay. And it matched what we observed in nature very, very closely. But this is a slide from Mark Rains. You know, Mark asked, well, if you put a particle of water in the bay, how long does it stay there? The E-folding time was 127 days in this simulation. That's the green line is the tracer mass as a function of time. And so when you stick something in the bay, and of course it depends where you put it, but in this case it was right in the middle of the bay, it's spread out, and that E-folding time is a fairly good estimate of how long stuff stays in the bay. Now, of course, biology and chemistry consumes nutrients. The nutrients do get recycled. And so, whereas we did not have that in the tracer model, the fact that it gets recycled means that this E-folding scale really does apply to nutrients as well. All right, well, we had some satellite images, which are the top panels. You can see the coloration and where the effluent effects on phytoplankton were the largest. And if you look down at the bottom panels, which are the simulated, they look identical. So the tracer model actually worked pretty good. Of course, you didn't need satellites, so the phytoplankton bloom initially was so large, you can just see it by eye. All right, Karenia brevis. All right, that's our next application. So what do we know? Well, first of all, it occurs naturally. And it's the bloom of a toxic dinoflagellate we call Karenia brevis. It generally initiates offshore under oligotrophic conditions. It's a little different from what you may hear because a lot of people say if we only would stop sprinkling fertilizer on our lawns, it would go away, which isn't true. It manifests as a nuisance once it gets transported and concentrated near shore where it kills fish. And both the offshore conditions and the transport are determined by the ocean circulation. So Karenia brevis ecology is not biology, it's a whole shebang, and the physics are as important to Karenia brevis ecology as the biology. Well, we don't know, do humans exacerbate it? Probably, but you know, we don't have a good handle on that really. And we don't really know what terminates a K. brevis bloom yet. So what happened in 2018? For those of you that were here, you'll recall it was probably as bad, if not worse, than 2005, which was really horrendous. And so two things were in play. First, there was a bloom in 17 that for some reason never went away. And it continued in bloom pretty much south of Venice, Florida. 2018 came along and the offshore conditions were conducive to a new bloom. And when that occurred, it added on to what was already here from 17. So we had that coalescence of two blooms, if you will, 17 and 18. The lower panel on the left shows cell counts as a function of time. beginning in October of 17, you see the 17 bloom, which never went away. And then it starts to jack up again as the 18 bloom came ashore. The red dots are when we saw a bloom on the Panhandle coast, and the blue dots are when we saw it on the East Coast. So the 18 bloom was an example when we had simultaneous blooms occurring on the west coast of Florida, the Panhandle, and the East Coast, all originating from the same place, namely the offshore region off of Tampa, a little bit north of Tampa. Okay, what did we do for 18? Well, we had a glider survey out there, and we saw elevated chlorophyll near the bottom, and in situ water collection and microscopy showed that Karenia was part of that. So we initialized our West Florida coastal ocean model at those locations, and then track where those particles would go. And that's the track lines that you see. The color coding is the depth. So it started deep, came up to the surface near shore. And this kind of demonstrates why the region Tampa Bay to Charlotte Harbor is the epicenter for Karenia brevis red tide on the west coast of Florida. 2020, 21. Well, 2020 was kind of a surprise. We didn't think we'd have a major bloom that year, but we did. It actually started off at Sanibel in December of 20. And then we watched it slowly move towards Tampa Bay. This may be an instance when human influence was in play. The Caloosahatchee River was peaking mid-October to mid-December with a big slug of water that came down mid-November. Was that the reason? I don't know, but we don't think this one originated offshore. We think this one actually was of local origin. And then along comes April of 21, and the winds were anomalously southerly that whole month. And we have this tracking tool both in our West Florida coastal ocean model and our Tampa Bay coastal ocean model. So we can actually track the movement of the Karenia as it slowly marched towards Tampa Bay. And once it got to the mouth of Tampa Bay, tides bring it in and slosh it back out. And around the end of May, we had this major bloom pop up right by Piney Point. So while pure conjecture, there is a smoking gun, and it might indicate that recycled nutrients, not the initial nutrients, but the recycled nutrients from Piney Point may have initiated that major bloom inside of Tampa Bay. Of course, once it's there, it kills fish, and those fish then supply the nutrients to sustain the bloom. And with our Tampa Bay coastal ocean model, then we go ahead and track once the bloom occurred. And it was a terrible mess. As you may remember, I ran away to Maine. I didn't want to stay here then. All right, so one more application. We got a call from the Longboat Key Police Department one day that a body had washed up by New Pass on the beach. And the question was, that person was last seen in the Intracoastal Waterway at a marina the evening before. Was it possible for that body to have been transported from point A to point B? Since the Tampa Bay Coastal Ocean model has very high resolution, it includes the inlets. We actually were able to do that simulation and demonstrate that it was indeed possible. And so that drowning was deemed to be an accident versus a homicide. And lastly, the flushing of Old Tampa Bay. Well, we know there are problems in Old Tampa Bay. To do a flushing experiment, you've got to have 3 dimensions and density dependence. You cannot do it with tides alone. Useless, worthless. And so we've done experiments with flushing with our Tampa Bay coastal ocean model. The other thing that we know is important is multiple inlets. If you have one inlet, it's not going to flush very well. If you have multiple inlets, it will. So let me leave it at that. And my concluding remarks are, we have this really nice tool. It's rather unique. It's the only one that downscales from the deep ocean right into the estuary. It has very high resolution, fully 3D, time dependent, available in real time on a daily basis. And I showed you a bunch of applications, and I'll try to take some questions if there's time. Thank you.
I think we're out of time, but if you have questions for Bob, you can catch up with him at the break, maybe? Okay, thanks, Bob. Okay, next we have Eric Muhlback. He is with the HAB group at FWRI, and he will be talking about dynamics of nanophytoplankton and other HABs in the Indian River Lagoon. So Eric is involved in many research fronts, but he coordinates the flow cytometry lab at FWRI, which is a tool that he'll be talking about today.
Thanks, Cary. The only thing I'd like to add to that is that the work I'm going to be presenting is the result of collaborative monitoring efforts with many partners, including St. Johns River Water Management District and the University of Florida. And Cary, myself, and the rest of the co-authors listed here just recently published this work in a special issue of Frontiers in Marine Science, if you're interested in learning more about this after the talk. The Indian River Lagoon is a hydrologically complex system on the east coast of Florida that consists of multiple subbasins and has been characterized as having long water residence times. The subbasins we'll be covering in today's talk are the Northern Indian River Lagoon, the Banana River Lagoon, and the Central Indian River Lagoon. These subbasins are influenced by multiple factors, including natural forces and disturbances, human population growth, and increased urbanization. Historically, this system has seen annual blooms of traditional HAB species, but since 2011, the system has experienced a shift towards blooms of smaller sized, ecosystem disruptive nanoplankton, including brown tides caused by Oreombra lagunensis, which we've already heard a bit about. It's these blooms that spurred us to begin high resolution monitoring efforts with all of our partners and volunteer networks in 2015. The specific HAB species I'll be covering today are Pyrodinium bahamense, which is a large armored dinoflagellate with a known resting cyst stage, and it's easy to identify via light microscopy due to its large size and distinct morphology. It's a traditional HAB species in the sense that it produces saxitoxin, which can cause paralytic shellfish poisoning. Pseudonychia is a marine diatom genus that consists of 59 species, and it's quite difficult to ID via light microscopy alone, so only genus level identification is possible. This species produces domoic acid, which some of these species produce domoic acid, which cause amnesic shellfish poisoning. Oreombra lagunensis is the southern brown tide organism, and it is a small, morphologically nondescript pelagophyte that's quite difficult to definitively identify via light microscopy. Though not a traditional HAB species, high biomass blooms of brown tide cause negative ecosystem impacts by decreasing light availability, causing trophic disruption, or creating fish kills when blooms crash rapidly and create low DO events like in 2016. Because of the challenges with IDing this species, we began using immunofluorescent flow cytometry and qPCR methods to complement our microscopy-based identification. So an antibody for brown tide for the immunofluorescence assay was originally developed in 1998 in response to a 7-year-long brown tide bloom event in Laguna Madre, Texas. After brown tide blooms later emerged in the Indian River Lagoon in 2012 and 2013, we began implementing and developing new tools for brown tide detection. The work was supported by the National Estuary Program in 2014, and we continued the monitoring efforts through the present day, documenting additional brown tide blooms in 2016, 2018 through 2019, and in 2020. The new tools we implemented were the species-specific flow cytometry antibody assay and a newly developed qPCR method developed at FWRI. So how did these methods perform? Here I have a bunch of regressions to kind of highlight that. But both methods, the flow and qPCR methods, are validated using light microscopy counts, which are mostly shown on the x-axis, including FWRI Oreombra-like nanoplankton cell counts, and also expert University of Florida counts, where extra care was taken by a very skilled taxonomist to provide a correct identification. Overall, the newer methods performed quite well across 5 orders of magnitude of cell concentrations. Flow cytometry compared to the light microscopy counts, which is shown in the top 2 plots, tended to slightly overestimate cell counts at low concentrations, but performed well at medium to high concentrations. qPCR compared to light microscopy, which is shown on the bottom left plot, tended to slightly underestimate counts at low concentrations, but performed well at medium to high concentrations. And flow cytometry compared to qPCR counts, which is shown in the bottom right, compared quite well, but we did have a lower N number here. Together, all data types give us a good picture of what's happening in the IRL, and can be used to cross-reference each other when there's anomalous counts or other questions that come up. Okay, so now we'll dive into some of the time series data, beginning with pyrodinium abundance trends in the IRL. Here and on the next several slides, I'll kind of have the same setup, where we have cell abundance and cells per liter on the y-axis, and the day of the year on the x-axis, spanning 5 different years from 2015 to 2020. The horizontal black lines will indicate bloom thresholds. Here for pyrodinium, it's 100,000 cells per liter, and the dashed vertical lines show the mid-year mark for each year. The color scales show salinity, and each plot corresponds to a specific subbasin, as indicated by the colored boxes and labels. Overall, pyrodinium exhibited a very strong seasonal pattern, with blooms generally developing in early summer and terminating in autumn. And this makes sense, as pyrodinium is a known seasonal bloomer that produces resting cysts upon bloom termination that overwinter until water temperatures warm again. Another interesting thing that we can see here is that the Northern Indian River Lagoon and the Banana River Lagoon have pyrodinium blooms more consistently than the Central Indian River. This is likely because pyrodinium is known to thrive in shallow, poorly flushed systems, and the Central Indian River has shorter water residence times than northern basins due to the presence of Sebastian Inlet, which you can see on the map. Lastly, another observation in the Banana River is that during 2018 and 2019, the typical seasonal pyrodinium blooms appear to have been suppressed and delayed in timing, and in 2019, bloom levels were not even reached. We'll circle back to this later when we look at the Oreombra time series, since coincidentally, these were years where there was an active brown tide bloom present in the Banana River. Oops. Oh, sorry. Moving on to Pseudonychia, we can see that this taxa bloomed more sporadically, both spatially and in timing, and was more broadly distributed throughout the different subbasins. In contrast to the pyrodinium blooms, the Central Indian River Lagoon appeared to have more consistent blooms of Pseudonychia than other subbasins, and this is consistent with Pseudonychia being a marine diatom genus that thrives in more well-mixed and saline environments. Okay. Lastly, we have the brown tide alga Oreombra lagunensis, where the bloom threshold here is much higher at 200 million cells per liter. And here we're showing both microscopy and the immunofluorescent flow cytometry data, shown as triangles and circles respectively. This species had blooms observed in all years and across all seasons, with very notable and prolonged blooms occurring in 2016 and 2018 through 2019. Like pyrodinium, this alga appeared to bloom more frequently and for longer periods of time in the northern basins, and recent papers have posed the possibility of this species also having a resting stage, which would make it well suited for persisting in areas with poor flushing rates. And although blooms did occur across all seasons, it's interesting to note that both the 2016 bloom and the 2018 blooms both kicked off late in the previous year, so in the winter time. And lastly, circling back to the Banana River Pyrodinium blooms that I mentioned earlier that appeared to be suppressed in 2018 and 2019, we can see that these years the system was impacted by a prolonged and high biomass bloom of brown tide, and modeling that we did suggested that the presence of brown tide had a significant and strong influence on suppressing Pyrodinium blooms during this time. So the year 2020 was special. It brought us a surprise bloom to the RL system. In early September, collaborative sampling involving many partners intensified in response to widespread accounts of bright green water discoloration. Efforts focused on following the progression of this bloom with satellite imagery, cell counts from light microscopy and flow cytometry, toxin analysis, and the determination of the molecular signal of this nano-sized phytoplankton, which was soon determined to be a novel cyanobacterium, the identity of which is still under investigation. Oops. At the height of the bloom, cell concentrations reached 3 billion cells per liter, and the bloom was sustained for almost 4 months with the most expansive spatial coverage stretching from north to south across all basins as shown on the bubble plot map here for the week of November 15th. The bloom finally subsided the last week of December, concurrent with a steep drop in water temperature. So looking at environmental parameters, here we have some kernel density plots showing how our different HAB blooms related to salinity and temperature, colored according to the legend where the cyanobacterium is in blue, Oreombra is in light blue, Pseudonychia is yellow, and Pyrodinium is red. Proportion of blooms is on the y-axis for both plots, and salinity and temperature values are on the x-axis. For salinity relationships, we can see that Pyrodinium and Pseudonychia blooms occurred at higher salinity regimes, and Oreombra and the new cyanobacterium blooms occurred at lower salinity regimes. For temperature relationships, Pyrodinium, Pseudonychia, and the new cyanobacterium skewed towards occurring during warmer temperatures, while Oreombra had a broad distribution but skewed towards cooler temperatures. So looking at salinity and temperature relationships combined on this plot, we can clearly see that each of these blooms occupy different salinity and temperature spaces with some overlaps. I have some kind of conclusion bullet points here with a few other points, but overall we can see that even when looking at bloom occurrences bounded only by 2 environmental parameters, distinct patterns can be found, indicating that IRL HAB species have distinct ecological niches and physiologies and/or life cycles that allow them to thrive when conditions and timing are just right. And this raises the question, have there been or will there be additional open niches for HABs in the Indian River Lagoon? 2020 surely brought us a new surprise, so hopefully our future work will allow us to better understand IRL HAB blooms and ecological niches to better predict and manage IRL HABs in the future. And with that, I'll just open the room up to any questions you might have if there's time.
We have time for one question.
Hi. Actually, maybe I should let Michelle
Oh.
or Melissa. Here, you go. Do you think it's possible that, so some of these insist in the sediment, correct? Is it possible that the seagrass loss is exacerbating the problem? Maybe the seagrass somehow consolidated the sediment such that it wasn't resuspended?
That is an interesting question, but I'm not sure that I can answer that. Yeah.
Maybe Natasha's is better.
That's an interesting thought, though.
Can you repeat the question?
I just have a general question. So I look at this kind of stuff when I do data QC and consider representative or non-representative for some of our routine monitoring. How are the thresholds determined? Are there certain things that are, because for the longest time I was just looking at Karenia brevis, so I'm kind of intrigued that now there are specific ones for the other ones. Can you just touch on that?
In general, the bigger size phytoplankton, you need less of them to create a high biomass bloom. Whereas the nanophytoplankton, there's that big difference from 100,000 cells per liter to 200,000 cells per mil. And it's mostly a biomass thing. I haven't set those exact parameters, but our folks and folks at the district kind of determined the one for Oreo umbra. And so that it relates to biomass essentially.
Thanks, Eric.
Thank you.
Okay, so this is me. So Eric talked about Pyrodinium and other HABs in the Indian River Lagoon. And so now I'm going to transition to talking about Pyrodinium bloom dynamics in Tampa Bay. So Eric gave a good background on Pyrodinium. I just want to recap a few of the characteristics that may give Pyrodinium a competitive advantage over some other algae in certain situations. First of all, Pyrodinium produces resting cysts, which are comparable to plant seeds in that they're deposited to the seafloor where they can overwinter and seed future blooms. And so this is important for Tampa Bay because germination of those cysts provides the influx of cells to the water column. Also, Pyrodinium is a dinoflagellate, so it can vertically migrate. It's a pretty strong swimmer for an algae. And so changing its vertical position in the water column can allow access to resources that may be limiting and not other algae may not be able to actively access. And then also the vertical position in the water column influences its transport dynamics. And Eric mentioned that Pyrodinium produces saxitoxins. Those are potent neurotoxins that can act to deter some grazers. And so this diagram, I just wanted to illustrate the complex dynamics of blooms and the different processes that interact in nonlinear ways. So from the let's see if I can get the pointer to work. I can't really, but so from on the left, you have the life cycle, which is important for bloom initiation, and then different environmental factors that influence cellular growth and loss, behavioral, physiological, and hydrodynamic aspects that influence bloom development. And then the bloom itself provides a positive feedback, seeding, forming more cysts that are deposited into the seed bed to seed future blooms. But today I'm just going to focus on a few aspects of this diagram. One, to talk about what are the drivers of recurring annual blooms in Tampa Bay, and then also to focus into 1 year in which we did high resolution sampling to take a look at Pyrodinium bloom development and decline. And so this graphic just shows Pyrodinium abundance in Tampa Bay every year since 2011, which is when FWC started its routine monitoring program. We know from EPC data, EPC routine monitoring and FWC opportunistic monitoring that prior to that, 20 of the last 22 years had bloom abundance of Pyrodinium recorded in Tampa Bay. But here I'm focusing on these 10 years to give you an idea of the spatial and the temporal distribution. So we have time on the x-axis as day of the year for each of the 10 years. On the x-axis, we have latitude from north to south in Tampa Bay with that dashed horizontal line indicating the southern extent of the Old Tampa Bay management segment. And then we have Pyrodinium abundance represented by the color of the circle, with the darker blue colors representing bloom abundance, which is over 100,000 cells per liter. So you can see that we have those bloom abundance, those dark blue circles every year, and they tend to peak in July, which is past that orange vertical line, which signals the mid-year. And then if we look to the map in the lower left, we can see the spatial distribution in a different way, represented as a frequency of bloom occurrence in routine samples. And so you can see that the larger circles are, which means more frequent bloom occurrence, occur north of the Howard Franklin Bridge. And when we look at the resting cyst abundance in the sediments shown in this map by the blue circles, we see a similar distribution with the highest concentrations of resting cysts north of the Howard Franklin Bridge. The largest blue circles here represent thousands of cysts per single gram of sediment. And this is important because of the life cycle, as I mentioned before, which is depicted in this diagram, in which the cysts germinate, a swimming cell emerges, begins to vegetatively reproduce, so cells divide, a bloom occurs, and then after the bloom, more cysts, the sexual reproduction phase is entered, more cysts are produced and are deposited to the sediments. When the cysts are produced, they're dormant and can't germinate. Our studies have also found that summer temperatures induce any ungerminated cysts in the sediments to re-enter dormancy, which effectively limits germination from occurring in the fall or the winter when the water column temperatures aren't optimal for Pyrodinium. And so that's what this plot shows. Oh, well, no, this plot shows that the cool temperatures of winter are required to break dormancy. So if you take, if you take dormant cysts and you store them at cool temperatures, when you get to almost 100 days, those dormant cysts will begin to germinate at about 50% rates upon incubation. So this seasonal modulation of germination and dormancy is important for the timing of germination and the influx of cells. But temperature is also important for the annual cycle of blooms because it influences growth rates. And so when we look at historical bloom data in Tampa Bay and the distribution in temperature space, we see that the majority of bloom occurrence occurs between 28 and 32 degrees Celsius. And so laboratory studies with a Pacific isolate have found that growth rates of Pyrodinium occur in a similar range. And so the annual cycle of temperature is important in multiple ways for the annual cycle of Pyrodinium blooms. Keep pointing at the wrong thing. So now I want to focus in on 2019, which we had a high resolution study and looked at both spatial and temporal resolution. We had 6 stations shown here in Old Tampa Bay in the vicinity of Bayside Bridge, within 5 kilometers of each other. And for that sampling, we had weekly sampling. We did surface mapping of chlorophyll A. We also did discrete surface and integrated water column sampling, as well as CTD vertical profiles and discrete vertical sampling of Pyrodinium abundance. We also had diel studies in which we looked at the vertical variability of Pyrodinium over the course of the day. I'm not going to talk about the vertical studies today. We feature, or we detail those results in our technical report. Today, I'm going to focus on results from the surface sampling. And we did find that trends and patterns that from surface samples were similar to trends and patterns that we identified in integrated water column samples. And so this is just a plot of the bloom data with Pyrodinium abundance on the top panel. And this is on a log scale, and chlorophyll A concentration in the bottom panel, also on a log scale. The different colors represent the different stations in that 5 kilometer area to capture spatial variability. And then I just have some horizontal lines indicating for Pyrodinium the bloom thresholds, and for chlorophyll 8.5 micrograms per liter is the lower one. And so with this weekly sampling, we were able to capture the bloom development in detail with the first bloom abundance recorded in mid-May. The bloom began to decline in mid-August and dissipated quickly with all samples below bloom concentrations by mid-September. Overall, the bloom lasted about 3 months. And I just wanted to point out the high spatial variability that we observed both in Pyrodinium abundance and in chlorophyll A. You can see that on some dates we saw as much as an order of magnitude variability. We also saw after the bloom dissipated that we still had elevated chlorophyll due to other species that increased in abundance after Pyrodinium had dissipated. So I just want to quickly go through this to talk about what we saw in the environmental parameters and in relationship to the bloom development. So we have the bloom on the top panel. When we look at dissolved inorganic nitrogen, which is in the second panel in green, we see that there was a dip in dissolved inorganic nitrogen, suggesting a drawdown during the bloom. When we look at total dissolved nitrogen, we don't see a specific pattern relative to the bloom. We do see a slight general increase in total dissolved nitrogen, but we don't see anything that points to drawdown of organic nitrogen. However, we didn't look at the different pools of the organic nitrogen pool, the different constituents. And then the bottom plot just shows light attenuation, and you can see that, and this is measured through vertical profiles, and you can see that we have high variability during the bloom, which reflects the spatial patchiness, and in general, it's elevated during the bloom compared to after. So we can then take those measured environmental parameters and join them in equations with physiological constants measured in laboratory experiments or from the literature to model light-limited and nitrogen-limited growth. Sugandha is going to talk more about these models and these results, but I just want to focus on 2 take-homes. One is that the nitrogen-limited growth rates were lower than the light-limited growth rates, which suggests that nitrogen was limiting Pyrodinium during bloom development in 2019. The second thing is that when we modeled nitrogen-limited growth rates, we used different half saturation constants. The ones shown here in this plot are the lower half saturation constants, which represent a higher affinity for inorganic nitrogen, and those most closely matched our observations, which are shown in teal, this time on a linear scale. And so that suggests that Pyrodinium may have a high affinity for inorganic nitrogen, and this is something we want to look at more. It could be that there's other nutrient flexibility that we didn't capture in our sampling, but this is a question we want to explore in laboratory experiments. And then when we look at bloom decline, as if you recall, we didn't see a notable decline in light or nitrogen resources that would have indicated a role in bloom decline and slower cellular growth. When we look at other environmental factors, we do see that at the time of bloom decline in August, we also saw a drop in salinity below this 18 PSU threshold. And that's important because when we look at historical bloom data and the distribution in salinity space shown on the right with the blue plot, most bloom occurrence occurs in Tampa Bay above that 18 PSU threshold. And this is supported well, this suggests that the lower salinity is physiologically not optimal for Pyrodinium and may affect growth. This is supported by laboratory experiments done with Pacific isolates, but we want to look at the local isolates and look to see how lower salinity affects growth, as well as how it might affect transitions, life cycle transitions. There could also be other co-occurring factors that are related to lower salinity, like different grazer populations. And so definitely something that we need to look into more detail. And so just to summarize our findings from 2019, surface samples for monitoring purposes to follow trends and patterns were representative overall of the water column abundance. Growth is likely nitrogen limited as opposed to light limited. And because our model growth rates, not considering any losses, we're very close to our bloom growth in the exponential phase, suggests that losses are low during bloom development, which suggests that actions that maybe target increasing cellular losses could have an effect on bloom development. And then finally, the cause of bloom decline is still unclear. Well, that's not finally, actually. We also want to look at more at the fine scale variability. So we can see that variability in this photograph so well. And the plot at the top is what it looks like when we map that as chlorophyll A. So we want to look at the underlying factors, and also note that when we went out on the plots on the left-hand side are on a neap tidal phase, and the plots on the right are a spring tidal phase. In general, that year we saw higher biomass on those neap tidal phases, so we want to look at what's driving that. So sorry, I'm at my time limit, and oh yes. So, and what room is that? Okay, down the hall. In Grand Central. Okay, there's 3 posters that we would like to gather feedback on potential additional management actions to address pyrodinium in Tampa Bay. So please go check those out, and please give feedback. You can give feedback virtually or with Post-its. So that would be great. Thanks. And maybe you can ask me questions then. So now we're going to go virtual, and I'll go ahead and introduce our next speaker. Dr. Sugandha Shankar will be talking about Pyrodinium bahamense growth and toxicity. Sugandha is also with the HAB group at FWRI, and she focuses on Pyrodinium and Pseudonychia physiology and toxicity.
Can you hear me now?
Yes.
Okay, sorry about that. So I'll continue the discussion about Pyrodinium bahamense, which is a toxic dinoflagellate, as you all know, and it is known for its bioluminescence, and it also forms annual harmful algal blooms in Tampa Bay. Next, please. Oh, sorry. So here is a map of Florida, and in the inset box, I have our study site, which is Old Tampa Bay. The blue circles show distribution of pyrodinium from 2011 and 2020 at our 9 regular sampling sites. And the sizes of circles are reflective of bloom abundance at those sites. Next, please. So we have already heard quite a bit about background of pyrodinium, but just to recap, these blooms can cause wide range of environmental health impacts. For example, in Tampa Bay, it has affected the seagrass recovery by degrading the water quality, and in other places, such as Costa Rica, saxitoxin produced by pyrodinium is known to affect wildlife. And these neurotoxins can also cause paralytic shellfish poisoning in humans. Therefore, it is important to design well-informed bloom mitigation and resource management strategies. Next, please. So in order to do that, we need to improve our understanding of the interactions that influence the bloom timing and location. I have this slide to emphasize how complex these interactions are. Keeping these complicated layers in mind, at FWC we have been focusing on different aspects of this bloom. Cary in her talk just went over some of her findings addressing the life cycle aspects, which is represented by a yellow box here. And tomorrow in shellfish session, Sara Kaminski will be discussing how grazing by shellfish may be helpful in bloom mitigation in Tampa Bay, which is shown in green circle here. And the focus of my talk is growth rate and toxin production by pyrodinium cells, and I will be discussing results from lab and field. Along with cell physiology, there are other questions too. For example, role vertical migration and hydrodynamics of an area can play in cell distribution and or accumulation. But in interest of time, I would not go into the details of that. Starting with lab observation, next please. Starting with the lab observations. So in lab, we looked at relationship between pyrodinium growth and irradiance levels. Light is an important parameter because pyrodinium cells, as has been previously discussed, can vertically migrate to different depths in water column. Also, when cysts germinate in spring, the vegetative cells start growing closer to the bottom where light levels may be low. So for our experiments, we had 10 different light levels, ranging from 10 micromole photons to 1400 micromole photons per meter square per second. And these were set at 1410 light dark cycle to mimic the photoperiod in nature. The photo over here shows an example of our experimental setup where we had 3 replicate culture flasks maintained at each of those light levels. Next, please. Using growth rates observed at the tested light levels, we were able to plot the growth irradiance curve for pyrodinium in lab. I do want to point out that these results are for an isolate from Indian River Lagoon, but due to similar latitude scale of Tampa Bay, we think response of an isolate from Tampa Bay will show similar pattern. We still have to conduct those experiments with Tampa Bay isolate though. In our experiments, culture did not show any growth below 60 micromole photons per meter square per second, and the observed maximum growth rate for our isolate was 0.26 per day. Next, please. The other two parameters that we estimated are the first was the compensation irradiance, which is the light level where photosynthesis becomes equal to respiration, which in this case is 130 micromole photons. So basically, average light in the water column will need to be higher than this for a positive growth rate. And then our next parameter was alpha, which was 0.002, and the alpha value is nothing but the initial slope of this growth irradiance curve, and it defines the efficiency of cells at maximizing growth in lower light conditions. So a higher alpha value suggests better adaptability at lower light levels. Next, please. So using these estimates calculated in lab, let's see where does pyrodinium stand in the phytoplankton world. Now, as you can see in this graph, in general, dinoflagellates are slow growers. And dinoflagellates are labeled as dino, and they're at the bottom left corner in this graph. Unfortunately, I cannot use a pointer to point it. But yeah, so they are slow growers and they're not well adapted for lower light conditions. And with respect to low light adaptability, pyrodinium seems to rank poor even among dinoflagellates. And I have marked our calculated parameters by the green and blue circles on the graph in the bottom corner. Next, please. So just to make the visualization even better, this graph compares alpha values for some of the dinoflagellates from literature. Next, please. And we see that pyrodinium sits here. So it ranks quite low when it comes to low light adaptability. So we can conclude that it is a slow grower and it is not a low light adapted species. Next, please. And here is a quick look at comparison of our isolate with the Malaysian isolate. And we see that our growth rates and requirements are similar to published values in literature for other pyrodinium isolates. Next, please. So using the values estimated in lab and observed population growth in field for 2019 Bloom, we were able to develop some simple models for hypothetical scenarios. Next, please. In the top plot, we have growth rates calculated using light-dependent parameters I just discussed and half saturation constants for some nitrogenous nutrients from literature for similarly sized dinoflagellate. And a glance at this plot suggests that growth did not seem light limited as purple dashed curve in the top plot is higher than other curves. The other three curves over here compare growth rates using different half saturation values from literature. And these suggest that pyrodinium growth could be nitrogen limited in Tampa Bay. Now, coming to the bottom plot, this plot compares our model growth curves with observed growth curve for 2019 Bloom, and Cary has already discussed this. Just to recap, the blue plot over here is the field data, and we can see that growth for models with lower half saturation constant are a better match as compared to the orange plot here, which uses a higher half saturation constant value. That is, models with higher affinity for nitrogen seem to be more accurate. Finally, the green dashed curve here shows the potential grazing may have in curbing Pyrodinium growth in Tampa Bay. Next, please. So before going into my toxin results, a brief introduction to these toxins. Saxotoxins are formed by a group of more than 50 known congeners, and they are divided into 3 groups based on their chemical structures. The first group is carbamate toxins, and these are most potent derivatives. Then we have decarbamoyl toxins with intermediate potency, and finally the least potent ones are N-sulfocarbamoyl toxins. Next, please. In Florida isolates of pyrodinium, we only find 3 of these congeners, 1 from each group. Next, please. And here is an example chromatogram showing peaks for congeners present in Florida isolates. As you will see, our results discussed over here are in saxotoxin equivalents, and these were calculated using the relative toxicities mentioned on this slide. And I also want to mention that our final toxin concentrations were calculated using values of B1 and saxotoxin only, as in most cases, decarb saxotoxin was below the level of detection. So next, please. So moving on to the results, the first panel over here shows changes in cell abundance and overall toxicity, and the second panel shows toxicity changes within a cell. Looking at the first plot, we see that total toxins in culture flask and also cell abundance showed similar pattern over the course of experiment. Then when we look at the second plot, which shows the changes within the cell, we see that along with changes in toxin per cell, we also see change in relative proportion of different congeners. Again, I have only shown the results for B1 and saxotoxin, as the third congener, decarb saxotoxin, was below the level of detection. Next, please. We have also analyzed some field data, but in interest of time, I'm just going to summarize our findings here. The chart here lists the average toxin in different kinds of samples. The most interesting point here is that cysts, or the resting stage of pyrodinium, is also toxic. There have been reports about non-toxic cysts for some other dinoflagellates in other areas of the country and world, but that doesn't seem to be the case for pyrodinium. So we do see toxic cysts in our area. And then the pie chart shows the relative proportion of toxins can vary in cells sampled at different times in lab, and in field it can vary for samples collected on same day but from different sites. This is interesting because, as you will recall, these congeners have different relative toxicities. So overall toxicity of cells can vary due to these changing relative proportions. Next, please. So now, just to conclude, what are the implications of our research for resource management? We have established that pyrodinium is a slow grower. What does that mean that grazing pressure can curb its growth? Then we also see toxins in cysts, so that can affect benthic feeders. And what does this all mean for oyster restoration in the area? I do not have good answers for these questions yet, as we are still exploring these questions. Next, please. And then just to conclude, some of the next step plans are to conduct nutrient and growth toxin experiments, and also to compare growth on different forms of nitrogen. So growth on dissolved inorganic nitrogen versus growth on dissolved organic nitrogen. And we are also in the process of conducting a thorough time series analysis of Old Tampa Bay data to better understand cell toxicity in field. Next, please. And finally, I just want to mention that this work would not have been possible without the teamwork of FWRI HAB staff, and people who played a critical role in data collection are highlighted here. And funding for this work was provided by TBERF and Florida Fish and Wildlife Conservation Commission. And with that, I will take any questions if there are any.
We have time for one question.
The silence tells me that I explained it really well.